Yes. I think I advanced your slide, actually, if you want to go back one. Sorry about that. Thank you. All right, I think we're gonna get going. Good morning, everybody. How are we doing? Day 2 of the Lakes and Rivers Convention. My name is Pat Goggin. I'll be your moderator and help keep our sessions rolling along. If you are hoping you're in the wildlife, fisheries, and natural history stream, you're in the right place. I'd ask you to please turn your cell phones down to silent or off if you could, and we will get going. We have a number of great topics and wonderful speakers for you. And to start things off, we have Gretchen Garros from UW Trout Station. She is an evolutionary ecologist that studies unique activities in aquatic vertebrates and strives to apply this knowledge to improve understanding of ecological and ecosystem change. Her talk is Zooplankton Tales of Long-Term Ecological Change. Please help me welcome Gretchen.
Thanks, Patrick, and thanks to all of you for coming out early this morning to join me to talk about one of my all-time favorite groups, which is the zooplankton of our lakes. I was really excited to get the request to talk about zooplankton kind of in a general way, but also to highlight some of the ongoing research and technology our science of zooplankton right now. So that's the goal for today's talk. Here we have a beautiful picture contributed by one of our artists in residence at the station, Rebecca Jabs, that highlights the beauty and diverse form of the group of zooplankton. So you can see some of the highly recognizable water trees here in the Daphnia, some copepods, and then some of the smaller Lepidoptera. And so oftentimes in lakes, you're going to be able to get rid of that.
That's what I'm trying to do for you. Yeah. Oh.
It should go away. Will it go away?
Anyway, zooplankton, as many folks know, are an intermediate in our food web, but many times they're black boxed or overlooked in terms of their diversity and ecosystem function. And so it's quite common when you look at a food web for zooplankton that you think of the nutrients and sunlight allowing for algal growth. The zooplankton are the primary feeders on that algae. And then they feed our small fishes, things like minnows, bluegill, and others, which then are the for our larger fishes. So this is the basic of the food web. And zooplankton, again, represented by a large Daphnia here, is a really taxonomically diverse group. In a single lake, you can find 40 to 50 species commonly of different types of zooplankton. And each one of those has a unique feeding strategy. If you do just the food web of zooplankton alone, it would be a diverse network of function that's just in this single layer of our lake. In addition, they are, so here's another way that we think about food webs. This is energy transfer in lakes. And so again, we've got a bunch of primary producers, the primary consumers. Not only are they feeding on that and transferring it then to our fishes, but they also cycle into what's called the decomposition or the recycling of nutrients, the microbial loop of our food web. So as those zooplankton eat algae, some of those zooplankton aren't fed on by fish. They die, they feed the bacteria in the lake, and that nutrient recycles in. And so this can all happen within the water column of the food web. I am going to focus today just on the plankton. Zooplankton, animal, so zoo meaning animal, and plankton meaning things that are in the water. There's a ton of other inverts, macroinvertebrates, larvae of insects that are in our lakes that have large contribution, but the plankton's enough for one 15-minute session. So we're going to target just these small microscopic organisms that are floating around in the water column and driving them. So
Don't know why that keeps coming back.
Okay, I lost my plan.
Okay.
So this is kind of the trophic pyramid, one that we often think about that's been brought through ecology in a number of ways. It gets you to think about primary producers here, they feed the herbivores, but it's interesting. If you just go out to a lake at a given time, your biomass may be distributed. But if you look at a lake throughout the entire year, you have a ton of algal biomass, a ton of zooplankton biomass. Those organisms are turning over on a weekly basis. Zooplankton, we're thinking of a 10-day life cycle to a month, sometimes a few months. So they're turning over multiple times throughout the year, but they're feeding our long-term lake residents and building up that biomass of fish stock, which is something that we manage heavily for and we think about a lot. So again, zooplankton, very important, in driving our food web. I'm gonna point at you.
Yep, sounds good.
And this is a diagram of the food web from Little Rock Lake, which is one of the long-term research lakes at the Trout Lake Station, home of a acidification project that went on back in the 70s, 80s, and 90s. And in this food web, there were about 92 species that were identified. And when you look at it, I think this diagram just really highlights the connectivity and the importance of zooplankton. Because down here you have your algae, so the different algal taxa that are growing, producing. Here's your zooplankton prey. This is, about 30 species of zooplankton. And if you note the interconnectedness with the different algal species, the interconnectedness with the fish and invertebrate predators that are feeding on those zooplankton, this ring is highly abundant and highly interconnected. That's really important for zooplankton. The diversity of your group of zooplankton in a lake means that if we lose one or two of those species, it might still be fully functional, it might still transfer the nutrients in a way. So there's a lot of redundancy built into the diversity of it. And then, we spend so much time thinking of fishes, but really the number and diversity in comparison to what we see in the plankton is much lower. So, next. So that was my kind of overview of why we should care about plankton. The next step is to introduce who they are. And so I know many of you in the room are well familiar with the different types of plankton, but I think there's also folks that maybe don't think about them as often. And so I'm going to go through the different taxa that are around it. So if you were in my classroom, I'd say these are the 3 words you got to know for knowing your zooplankton, right? And so those words are rotifer, cladocera, and copepod. Those are the 3 main groups of plankton. If you look at them under the microscope, those are the 3 that you should be readily able to identify the difference between. And so we're going to dive into each of these groups, and I'll have some details about them, some research that's taking place on the specific groups. But I also want to highlight some of the biases that we carry from our past understanding into the modern way that we think about lake food web. So let's go to the next one. So we're going to start with rotifers because no one's sketched. So rotifers are this incredibly diverse, gorgeous group of zooplankton, but they're tiny. They're ranging in size from 10 to 40 microns. It's kind of a general. They can get up to 80 microns, but they're very, very small compared to many of the other groups of plankton. But here are just a few examples. Some of my favorites, Calabucha with these long spines that limit some of the feeding on it. Gastricus, when it is preserved in ethanol, looks like a tiny bag of guts, but when it's alive, it has this great rotating band of cilia and structure that helps it to move around in the water column. Splanca, it actually eats other plankton. So this rotifer is one of the biggest ones. It eats other rotifers. It can eat Cladocera, kind of a fun one. Again, when you preserve it in ethanol, it turns into a transparent bag of guts. It's hard to identify. And then I think this is a Brachionis down here. The thing that defines rotifers is the rotifer structure. It's the band of cilia that's at the top. That's called a corona. And it's basically these little cilia that whip around and help to pull in algal particles for their feeding. And the other structure is nicely apparent on this image. Right here, it's called a mastax, and it's these hard structures that sit there and go like this to grind up the algae or bacteria that they're eating and process those nutrients. And so while they're when they're preserved, those are 2 of the main structures, this feeding structure and the band of cilia that helps us to identify, as long as what we call the toe or the foot of the organism helps us to identify our rotifer groups. They can live solitary, like the ones pictured here, or this is a conopilus that is in a big ball. We find this in tons of lakes up north. It can be highly, highly abundant, tens of thousands in a single liter of water. And I showed you those beautiful pictures, but in reality, when you're thinking of rotifers, this is why no one thinks of rotifers first. This is what they look like under a microscope at a low magnification. And so highly hard to identify. It takes a lot of time and expertise to kind of get narrowed down into what they look like. And this is a legit picture of any plankton counter in the room knows that is what we really look at. And so those pictures are really quite captures. Go ahead. And just a fun fact about rotifers, I think something people don't always know, is that their evolutionary history, they're actually in a completely separate phylum from the other zooplankton. So they are not crustaceans. Daphnia and copepods are both in the group Crustacea. They mold their exoskeleton on a regular basis, but rotifer are a phylum completely of their own that's more closely related to mollusks and annelids. And so up here, can you tell which ones are the rotifers and which ones are mollusk annelid?
Mouse on the right.
So this is probably the most readily recognizable to folks working in lakes around here. This is a veliger larvae. It's got these lobed cilia for feeding, but this is the start of a snail shell. And so many of our bivalves in freshwater have a veliger larvae. And this is the lobed feeding structure of a rotifer. You can see how close those look to each other, and it shows some of that evidence. Oftentimes it's the early developmental stages. And then this is a polychaete worm. It's marine, but you can see some of the similarities between that and a common freshwater rotifer called polychaete with its structures and its structure. So good to know that these guys aren't crustacea, phyla all of their own, with a couple thousand species within. And like I said, I think rotifers are underappreciated both in their diversity, but also their function. From the long-term ecological research program, so Trout Lake Station and the Center for Limnology in Madison are home to a 40-year long-term ecological research program that is supported by the National Science Foundation. And that data set has been collected continuously since 1981. It includes plankton samples that have been taken every 2 weeks during open water and about monthly throughout the winter, although we avoid some of the dangerous shoulder season periods. So using those data, I dug back in to look at the relative effect of rotifers in our northern lakes. Up there, we're using 53 micron nets, so we have a pretty good representation of these small organisms, but we're probably still missing some of the smallest of the rotifers in our collections. But here are the records from, this is basically 2008 to, oh, that's supposed to be, I think it's the 2008 to 2018, so it's a 10-year window of time. And we basically averaged the abundance of the different taxa for each month throughout the year. And then we use published grazing rates for different taxa of rotifers, podocerans, and copepods to ask what is the relative water clearing taking place from these different taxa. And so up on the top is the relative abundance. And what you can, this is just number, right? We've got tiny rotifers, but we have hundreds of times more tiny rotifers than we do podocera or copepods in the water. And that proportional difference, when you break that down into clearance rates of these different groups, what you also see is that rotifers are making up about 50% or more of the grazing pressure in our lakes compared to things that get a lot of attention like Daphnia and copepod. And so the gray bars here are rotifers. Here's all rotifer with a few Daphnia and a few copepod. Here you can see that the small number of Daphnia and podocera do graze a lot. That's the yellow and the brown here. But still, the rotifers are a big contributing factor to what we should be thinking about when we think about carbon transfer and where all of that energy in this rotifer group is going. But it doesn't come into models as commonly, and because it is hard data to collect and process, can be overlooked. The other thing to note is that many management policies, our LTR program down in Madison, are using 80 micron nets. The rotifer groups are mostly below that size class. And so I did try to compare north and south within our LTR data, but the rotifers are woefully underrepresented in that data set where the larger mesh is used. And so this is something to consider if we want rotifers to be part of the stories that we're creating and the ecosystem management that we're doing. We also need to consider that sampling and how we process and bring them in. So there's a lot of push for more inclusive zooplankton counting, keeping more of these represented. So, oh, this is similar data. I just wanted to show that the long-term part of this. So this is that long-term data set from the 80s up into the 2020s. And again, you see that rotifers, so this is the grazing or the water filtration. Again, it's not just a one-off event where they're showing up. They are a big part of grazing over time. We can also look at how their relative effects and how other species within the systems change over time. And I keep saying rotifers. I'm doing exactly what I said I wanted folks to kind of avoid, but species are also important. We've got this diversity of rotifers. I'm grouping it all as just rotifer. But I liked this. This is a long-term, a long ago study based on data from the 1940s, so published in the 1980s. But it's a really nice kind of iconic representation of how even within a single genus, these are Polyarthra. They're the, I think they look like, I don't know why, but I think they look like those little barrel candies, like the brown, they kind of get a brown color, and the mammal hair is coming off. So I think of them like those barrel candies that you maybe didn't want to eat or you love them or whatever. But Polyarthra, they have a bunch of different species. And so here's an example, which is 4 species where they're feeding it Chlamydomonas, which is kind of a perfect little algal food for most zooplankton. This guy up here, just a big sack of green goodness. And what you see is that even within this single genus, they have different feeding preferences. One comes in early and is really successful, but as others come in to compete, you get a generalist and then vulgaris here. And then you have transitions into 2 other groups throughout the resource availability. And so you see these sequential events, even within just this one genus of the rotifers. And so the species transitions that take place throughout the season can be substantial among those 50 taxa that you might find in the lake. All right, so now on to something folks probably do recognize readily, and that is the Daphnia. And not to do it disservice, I will talk about how it has been blown up as a model organism and is something we readily recognize and put into our lake thinking. I think that's a valid thing because they have a disproportionate impact on the lakes. But I do think it's important to consider other classes like the mighty Bosmina, which is tiny but can be incredibly abundant. It looks like a little elephant when you see it. It's got this long rostrum that curves around its body. And these guys are found in so many bodies of water, almost everyone. They love little ponds, but you can also find them in shallow lakes and large lakes. Hydorids, so lots of different Cladocera. These are known for having a carapace that wraps around their entire body. And so their little legs and identifying features are kind of in that transparent structure. And the interesting, go for it. So there's lots of interesting things about Cladocera. One is that they undergo what's called a boom and bust strategy. So the Daphnia in particular are generalist feeders. They eat almost anything, except they don't love toxic cyanobacteria. And so they'll spit out all their food if they get into a patch of fixed cyanobacteria. But they eat all the other algae and they can do a pretty good job of it. And so this is a data set. This is from Lake Mendota in the 1980s, 1970s. And the black line here is the Daphnia peaks. And so they come in spring following the big algal bloom, and then they eat themselves out of house and home and just drop off precipitously. So this boom and bust is a dynamic that's known in eutrophic lakes. In large eutrophic lakes like Mendota has been kind of an iconic representation of that, and it leads to what we call the clear water phase. They eat all the algae, and folks get really happy. The water often will have this phase of very clear water before it then begins to accumulate other types of algae with the Daphnia phyllowite. So go for it.
Sorry.
Ready for next. So the reason that this happens is because of the unique life cycle of the Daphnia. And so Daphnia are cyclic parthenogens. What that basically means is that they alternate between asexual reproduction, where females produce clones of themselves rapidly, and sexual reproduction, which produces a long-lived dormant stage. And so during this phase of parthenogenesis, the female can produce anywhere from 10 to sometimes up to 50 eggs for some of the larger Daphnia species. And when the food is good and they're producing 10 to 50 eggs in a single 10-day generation period, it means that their numbers can go from here to here, and they're all genetically very similar, right? They're clones of their mother, genetically identical. So this asexual reproduction leads to a monoculture of highly efficient feeders in that exact environment. And that strategy is the one that can lead to what we call exponential growth, right? Just doubling, tripling, quadrupling of the population each 10 days or 20 days. And that is what drives this boom and bust dynamics in our lakes when the algal conditions are good. So this life cycle is also, this life cycle is also what makes Daphnia what we call a model research organism. We've chosen model research organisms in many groups. You think of lab rat, right? Everybody thinks the lab rat's a model organism. Frogs are considered a model organism. In the plant world, it's ragweed, Arabidopsis. And there's reasons that this happens, where we drive our kind of inquiry based on the characteristics of the organism. The other reason that it happens is because a long time ago, people decided to work on it. So Daphnia experiments have been going on for 120 years in academic institutions. And it's because of some of these characteristics that it was brought in. It eats anything, it grows really fast, it's pretty easy to keep in culture, and it produces clones, which allows you to take the exact same genetics of an organism and distribute it into different environments and test how it responds to them. These are characteristics that are great for a model organism, and just the longevity and the understanding that people and efforts people put in early on this group. And so the studies that have come out of Daphnia research are incredible and far-reaching. They work for lake biologists, but they've expanded into basic understanding of evolution and development. And so I do agree that it's a model organism and one that draws attention to the lakes. But when we go into ecosystem thinking and food web thinking, it's not. Go for it. So it's more Daphnia stories. My origin was working on Daphnia dormancy. I was an undergraduate at Lawrence University in 94 to 98. And I spent 2 summers at Trout Lake working on dormancy and how the diapause eggs were allowing for recovery from the lake acidification experiment that had taken place on Little Rock Lake. I'm not going to show those data today, but I am going to emphasize the usefulness of this dormant stage. So keep in mind that this is the sexual phase of Cladocer's life cycle, which means that it's genetic recombination, but it also allows for dispersal in both time and space. And we think about it in terms of space, right? We've got the Clean Boats Act, where we're washing off boats and trying to get these diapause eggs off the boats so we're not spreading spiny water flea around, but also thinking about it in terms of time. For a 10-day or a month-long life cycle in the water column, some of these eggs can last for up to hundreds of years in the water column. And can you imagine if the genetics from 300 years ago in Lake Mendota, if you had an organism that lived there 300 years ago, hatched out today? What is going on, right? Even trout lake, I think about, which has had change in the last 300 years, pretty substantial. Just coming out and thinking, oh, this environment is completely different. So go for it. So it's a little way full time capsule of genetics and history. And we get to use them that way in aquatic research. And so there's this field called resurrection ecology, which means that you can go in and actually use these diapause eggs, you can hatch them out from past time periods, and you can look at how they've adapted to conditions over time. So this is an iconic study by Nelson Hurston Jr. et al. And they were working in Lake Constance in Switzerland, and that lake in the 1970s became really polluted with phosphorus, and there was a ton of primary productivity, which led to cyanobacteria and what we call bad algae growing in that lake. And so what they did is they dug into the sediments and they pulled out a bunch of eggs out of that lake, and they hatched out clones from the pre-pollution period. They then went in and took eggs right during, right post, kind of right during that high pollution period, or when that had been happening, where the food was going really bad in the lake at that time. And then, and this, was published in 2001. So then the kind of after the lake began to clean up and there was more good algae around. And they grew those the same. So they grew up those clones. They had about 10 clones from each time period, representing what was in the population at that time. And what you see is in the early days, nothing had adapted to bad food, right? If there was bad food, these guys were going to have a really bad growth rate, and they were not going to be able to compete in the way that or survive in the way they would on good food. And so this is called phenotypic plasticity. It's a reaction norm. So we think about evolution in terms of how well do things respond to good food versus bad food in a scenario. So they had a lot of flexibility, but it could go really poorly. No one wants to grow rate this low. It's not going to lead to health. So after the stretch of bad food, these Daphnia populations had evolved to be able to deal with that bad food. They could grow and succeed on bad food. So the slope of that reaction norm, you no longer have bad food killing off a bunch of your Daphnia clones. Now, all of the ones that are left are adapted to survive on bad food. As the bad food disappears and the lake conditions improve, we see that selective pressure for survivorship in both conditions change, and with the kind of restoration of that lake, they're evolving to where they're a little bit more flexible again. And so resurrection ecology actually allows us to dive in and ask, how does evolutionary change impact the adaptation to the ecological changes and the environmental changes that we're dealing with? And so it pairs those two units in a really cool way. Daphnia. This I just added because I think it's amazing to keep context. The other thing to note, and something that we also don't always reflect on when we think of freshwater zooplankton, but these lineages originally evolved from marine lineage. And cladocerans in particular evolved into freshwater and then diversified from there. Copepods, we've had a number of different species of copepods that have repeatedly come into freshwater. Rotifers too, they come into freshwater. But then the cladocerans, once they reached freshwater, they branched into all different types of groups. And so with the Daphnia, we also have the entire genome that's been categorized for 2 different species, Daphnia pulex and Daphnia magna now. And with that, we're able to trace the family tree of the different Daphnia lineages. And so this is a bunch of different Daphnia species that happen throughout the world. And I just think it's fun. This is a family tree or phylogenetic tree based on DNA. And what you see is that there's this origin of Daphnia way back here, but then it quickly broke into what we call the North American clade and the Eurasian, or the Eurasian Australian clade. And then once it got to North America, which includes all of North America and quite a bit of South America in this clade, Daphnia, different populations, you see that most of the major groups that you would recognize from your plankton counts are present here and have diversified within the waters of North America. So the taxa that we have are adapted for these waters. They evolved here, they've adapted to these waters. And that's why we would consider cladocerans that may be introduced into these waters. It creates a slightly different story, and they're going to have different impacts. They're also potentially going to have challenges when they try to establish and gain kind of diversification and function in those systems. The other thing for a plankton biologist is that if you go to Australia, you're not going to know anything that it's only taxa, which is a super fun challenge, but it is interesting to think about the fact that really the species that I know and love well, I feel a little out of luck when you get into another region. So they are diverse, they're changing based on the immediate environments that they're living within. Copepods. Copepods are another group of zooplankton. They get a ton of emphasis in the marine system. This is one of the major players in marine system. A lot of folks don't realize that the copepod daily vertical migration in the oceans is considered the largest biomass migration on Earth. Then in the krill that are moving up and down hundreds of meters in the water column on a daily basis. And so they are highly abundant, a little less diverse than the rotifers, but still big players within our system. And you don't have to outline all these names, but there are two major groups, the calanoids and the cyclopoids. And then they have the larval stages, which are nauplii. So if you're looking at a plankton sample, often you'll find many, many nauplii. They're hard to identify as species, but once they get into their adult phases, the calanoids are famous for their extra long antennae, and the cyclopoids have kind of these shorter, stubby, curlier. So copepods also have long-lived dormant stages, and they have the record for the oldest dormant stage. So 332-year-old egg attached out of Bullhead Pond in Rhode Island back in the 90s. And I just think this is a cool picture to think about the development of this diapause egg as it comes out and forms this little spider-looking copepod called the nauplii stage, which again can be highly abundant and effective grazers. The other cool thing about these eggs is they can dry out, they can be in full sunlight for a long period of time. They can pass through the guts of frogs and fish and birds and stay viable. And so they are great dispersal units for our zooplankton residents and copepods. Even though Daphne and other Pledosphera have diapause eggs, copepods are usually, if you have an open water body, copepods are usually the first ones to get there. They get there, they start breeding. Go for it. Yeah, so I mentioned that they are one of the groups that has been studied heavily in marine systems for vertical migration. A little bit about vertical migration. I'm going to move into a research vignette about this that we're working on. It's just the principle that some plankton will move up in the water column at night to feed on all the good algae that's been growing up there during the daytime. And then during the daytime, they'll move down into the deeper waters below that thermocline, below into the cool waters. And most of that is to avoid fish predation or visual predator predation up in the surface waters during the daytime when light's available. And then they'll migrate back up to the surface as night comes on to play. So this DVM behavior is one that's well documented for copepods and Daphnia, not as well for rotifers, but probably happening for some of them. So next. I had to highlight a gorgeous picture by Christina Weatherford, which was a SciComm intern at Trolley Station this last summer. I think it's coming on to the UW College of Letters and Science back page that's coming out. And it was around our vertical migration work. And so here's the Milky Way and all of these little zoops coming up at night to eat all the great algae that's there. So I'm going to go into a little bit of a research vignette. A research vignette. So over the last 2 years, I've been collaborating with Mark Stasio and from Lawrence University and a couple of student interns each summer to revisit some of our assumptions about vertical migration. That diagram I showed you is kind of a straightforward, simple diagram of how we think it works. But as we dove into different types of lakes and different types of organisms, of course, the story is not as clear as that beautiful straight line that was showed for the copepods in the last figure. So the two questions I'm going to highlight today are one, how does DVM compare across lakes with different productivity and different depth structures? And then how does night light influence this behavior? Can a full moon keep everything suppressed? So this is Eleanor Meng and Reese Lavejo, who were the students on the project last year, and they took vertical migration samples from 3 different lakes: Renly, which is kind of down south, a shallower, high productivity lake; Big Muskie, which is by Trout Lake Station in Vilas County; and Sparkling, which is off of by Trout Lake Station in Vilas County. And the way that we do this is we take out, she's got the Schindler over the side, we take out this box trap called a Schindler trap, and it allows you to lower it into the water column and take a sample of just a single meter square of the water that's in the lake. So you can sample 1 meter, 2 meter, 3 meter, And rather than a plankton toe, which integrates the entire water column, this allows you to take specific depths of sample. So we did that in the daytime and at nighttime and counted daphnia densities at these different periods. And Bradley is probably the most apparent here. You can see that during the daytime, the daphnia are down in the deeper water. At nighttime, we still have them down at the deep, where we see many more of them up in the shallows. So we do have migration of the daphnia moving up, but not all of them, which is a continuous theme in our deviative measurements, even though we would assume they move up and down. And so again, in Big Muskie, we see that they're kind of distributed through the day, but not tons right up the top. But we do have some migration upwards into these waters, but none that go right up to the surface, on that very surface water layer. And sparkling also, we see at night more zooplankton up in the shallows, but not a ton of distribution once you get down below the thermal line. So these three lakes let us know that the temperature thermal line, the zooplankton really are hanging out below that during the daytime, but they're not pushing all the way down to the water. There's probably some energetics that go into that. That deepest water is full. Why swim farther than you need to if you're in that reservoir from the fishes? Maybe they're just going down where not as far as they need, but no farther. The other thing is there's a chlorophyll layer that accumulates at the thermal line. We call it the deep chlorophyll maximum, and it's this weird layer of chlorophyll that gets stuck at that boundary. And it could be that the zooplankton are going down to the bottom of that and just eating there for the daytime, and then they hang out right around that layer. There's still foraging capacity, and so that's actually where we're headed this summer on the project, is inspecting that deep chlorophyll maximum and this deeper distribution of the zooplankton. But the other things to note is Renly has the highest productivity, and shallowest lake did show more vertical migration than the other lakes. And the other thing that we weren't looking for, but it keeps coming out, and it's something that people are catching on to, is that overall the density that night were higher, right? Those black bars, you look at that and you say, well, you got just as many down deep, the distribution, but look at how many more zooplankton are being captured at night. That is something, next slide. That is something that Sadie Bullock-Farb and Ms. Jeff Ubik, two students that are part of the Global Lakes Ecological Observatory Network, the Leon Network, have also caught on to. Jason Stockwell is a zooplankton ecologist, and in discussions at that big global meeting, people are seeing this all over the place. Nighttime samples have more zooplankton than daytime samples. And this is in those vertical haul nets in the center of the lake, which is where most of us see our zooplankton samples. And so these are some figures from an analysis of about 230 lakes that they did globally. And it shows this is the percent change at night. So if it's more than 0, it means the number of zooplankton at night went up. And these are different taxa. But the real point is, here's the 0 line. You don't see a lot of decrease at night. And in some cases, you see 1,000% change in the number of zooplankton that you're capturing in the same body of water and using the exact same tool at the same location at night than you do in the day. When do we estimate our zooplankton in lakes for ecosystem modeling and monitoring? Not judgment, but this is science, right? We are missing potentially 1,000% of biomass of zooplankton in our lakes. That is crazy, and I feel responsibility, but I also feel like there's an opportunity here to expand our knowledge and to understand. So where are those coming from? The night zooplankton. So for a long time we talked about diurnal vertical migration, but we also need to be talking about horizontal migration. Macrophytes in the shallows provide a lot of hiding habitat for small fishes. The zooplankton are probably just as readily using that. We know horizontal movement happens, but is it able to explain this magnitude? The other part is sediment water coupling. So we also know if you get down into the mud, you can find zooplankton that are right at that boundary. If you ever snorkel or dive in the bottom of the lake, you can actually see them kind of chilling out right there on the bottom. And so there could be this demersal plankton eruption at night or these horizontals coming in. but it is important to know that this is happening, so. Cool, next. All right, and moonlight does not seem to affect diver migration. This was a fun study. Some of you probably met Julia Westberg last night. She's running a boat program now down in Madison. We're super excited to see that step in her career. She was a freshman intern with me in 2019, and we went out to Crystal Lake and we sampled every other night for an entire lunar cycle, thinking that maybe the bright moon in the sky would drive the plankton further down, and that migration might not be as substantial on a moonlit night. But our data do not support that. In Crystal Lake, they're interested in exploring in a lake that has a little bit more migration in general. Crystal is kind of strange when it's got a bunch of smell and it's not very highly productive. I'd like to try in Brenly, that lake where we see a lot of migration. But one thing that did come out of our work together was that the different taxa, so here's our rotifers, scopopods, and podacera, their rotifers are hanging out down deep. They're just really sticking down there every night. They're not moving up super high, so this is their average depth in the water column. So there are some distributed, but they're definitely lower down than the other taxa. Podacera at night are kind of in this mid-water. Here's our front line and the red line. And those calanoids are just hanging out right at that line. They're not even going up into the surface waters in time. These guys are, they are moving up and down, but I thought it was interesting how distinct the different taxa kind of spread out and use the water column primarily. There's definitely overlap. These are just averages, but it is interesting to think that among these three groups, we see some differentiation. Next slide. Research vignette 2 is transitioning away from kind of our standard PEG model. So a while back, the conceptual PEG model was put out there. Models are really useful for being able to discuss and predict and think about what's happening in systems. But I think the most interesting thing about models is when you deviate from them and you need to explain why. And so the traditional model for zooplankton understanding is the phytoplankton boom. The zooplankton then follow that phytoplankton boom, and then there's that bust that happens. And so as that bust happens, then the algae transition to less edible forms of algae. They stick throughout the season, and things like cyclopoids and calanoids and rotifers that just eat specific species do pretty well. They can cherry pick the ones that are more edible, but you get your big nasty algal blooms. You get, this transition and just kind of the slower meandering species assemblages of. So this is one of the models that is inherent in the knowledge of textbooks and thought about, but the deviations from this PEG model, they're becoming more apparent and obvious. And I think this is especially apparent in some of the work we've been doing under ice and with seasonal changes that are happening. And so here we've got that same rotifer curve. But what I wanted to highlight is these are winter, mostly Iceland months where the zooplankton numbers are low. They're present, they're playing along. I honestly figured didn't even have one about it. And also that boom and bust isn't always true. So in some of our lakes. So next slide. So we've got a couple of projects to tackle this. One was led by Ellie Socha, who was a graduate student at the Center for Limnology. And she worked with Hillary Dugan to clear off an entire bog lake for 2 consecutive winters and look at how snowpack was influencing the plankton dynamics under that ice. And so here's a picture of Ellie with her plankton net. That's where I came in to help them with methods and they were trained up on counting. And here's them snow blowing the entire surface of the bog lake to lessen the snowpack. Okay. And her results just came out this year, and we have a reference year back in 2019 where the snow remained on the lake, and then we had the 2 years where the ice was or the snow was removed. And what happened is in the first year, the ice came on and then it snowed really heavily, and so we got a thin layer of clear ice and a ton of what we call white ice. White ice does not let as much light through, and it's a little bit different than having that clear layer of ice. In 2021, the ice set pretty thickly, got a lot of clear ice. Here it's called black ice. It looks kind of black when you put it in the water. That clear ice, and then just a little bit of white ice on top. So one of the things that came out of her study is that snow and ice are not just snow and ice. It actually depends on how they all set and fall. But what you see is when you have a lot of clear ice, you get a lot of phytoplankton. Not as much when you have a lot of snow and a lot of white ice. So that clear ice had a lot of phytoplankton. We did actually see an increase in the chlorophyll, or a lot of light. We did see the increase in the phytoplankton chlorophyll count in both of the snow removal years. Next. And our algal populations showed especially large changes in the dark or the clear ice here. So again, these are the 3 years. Here's the algal population, the phytoplankton population in that clearance year. And you see that substantial increase in primary productivity when you have more light available, which is great to know, but really prefer to actual conditions that are happening. Right? We have years where we have a lot of snow, like this one, and years where we don't. And so the altitudes change, which means the zooplankton are changing.
Come on, no,
Oh, it's getting colder.
I'm sorry, yeah, you might have to go back. Can you go back?
No, it's not.
That's the right one, okay.
And so the rotifers, these are all pretty much rotifers that happen in this lake. It's a bog lake. The rotifers also increased drastically. There were rotifers present in almost undetectable numbers before, but the numbers of rotifers skyrocketed in the high productivity year, more oxygen available, more resource available. But the other thing to note is the species changed substantially. We went from kind of this diverse assemblage of different types of rotifers to dominated by Ascomorpha, which is a little transparent sucker guts. It eats, that's very efficient at eating small algae, and a Zooplankton, that predatory rotifer that was probably eating all that Zooplankton. And so the species changed with this seasonal dynamic as well as the vignettes. Next. So the take home on this is under ice environments lead to species specific changes that can then set up the stage for our summer dynamics, which was often the focus of that work. So next. The other kind of seasonal change dynamic that we think about takes place in thinking about ice off. So this is something that I've been talking about a lot for the last couple of years. We all recognize that ice is being lost from our lakes, right? And this is the graph that shows that long, slow change where we've lost, up to 20 days of ice in the southern part of our state, and we're losing ice about, a day or two every 10 years is about the rate that we're losing that ice continuously. But just as interesting is that in the next, what's as interesting, this is the variance in ice off date, is that for a long time it varied, right? Year to year it would vary. But our back-to-back ice off date, the variance in that has increased 5-fold in the last 10 years. And we feel that in 2020, ice off was in March, mid-March. In 2021, it was late April. There was at least a 30-day difference just between those 2 years. 2012 and 2013, there was actually a 52-day difference between ice off days. Think about a 10-day plankton living in these habitats. That's many generations and exponential population growth. So this is impacting the food web in general. So next. And here's just some examples. So phytoplankton, their vector is early ice off. When that happens, the ice goes out, the plankton grows early, the plankton grows late. The zooplankton are following suit. We get lots of plankton in those early ice off years following the zooplankton boom. But in late ice off years, it's not the same as much. And so lots of research going into the seasonal dynamics of zooplankton, how that's then feeding into the food webs. And when you think about not having enough zooplankton in the population, during that spring window, the transition of all of our spawning fishes requires that zooplankton. I'm going to pause here with my research vignette. Sounds like I'm close to time. And so I would be thrilled to take any questions.
If we have time for 1 or 2 questions, please. I think we have one from online. If we have no takers in the room.
I gotta see my go for it.
Does the type of lake, whether it's like a clear lake or a stained lake, dictate what type of zooplankton you're more likely to have?
It does, and those also have different dissolved organic carbon structures that kind of drive the primary productivity in those lakes. And so you often do find different taxa rotifers especially will differ between those lakes. You can also find that, sometimes the Daphne and the larger zooplankton will have, pigments, so that they're, or they'll carry hemoglobin, which allows them to have more oxygen in the brown lakes that we red. And so you see both changes in the community composition, but when there are overlapping taxa, you can sometimes see changes in their body structures. So, yeah, definitely influence that, both in terms of where the carbon's coming from the food web, and also just the defense against predators and some of those food web things.
Maybe time for two online questions. Are diapause eggs the eggs of Daphne and sexual reproduction?
There,
They are.
yep, genetic recombination dispersal in time and space.
Thank you. And does DVM occur more at different times of the year, or is it related to other sedimentary changes like bug head?
That is a great question, and I worked in the Mississippi for a long time, and we did see changes in the plankton distributions with some of those massive patches that took place in the river. I don't know the answer for that in lakes as much, but it is a really thoughtful question. Definitely more when the ice isn't on, because light matters for the day over for migration, but I don't have a straight up answer in terms of that.
Well, hey, thank you, Gretchen, for getting us off to a great start here. Thank you for the online folks who joined us as well. I hope you had a good experience. Thank you for getting through our little Zoom glitches too, folks. Next up at 9 o'clock, we have a plenary session and digital production that we always do. So please join us there in the plenary room at 9 o'clock. Thank you, folks. Thank you, Cam. So we will put